Last time I had discussed Clostridium tetani and Clostridium botulinum. Today I will start with Clostridium perfringens and Clostridium difficile. These are your learning objectives. You should know mode of transmission, pathogenesis, clinical features, and laboratory diagnosis of Clostridium perfringens in detail. And you should especially focus on clinical features of Clostridium difficile. Clostridium perfringens, so it's an obligate gram-positive rod. Spores are present in soil, though these are members of normal flora of colon and vagina. Clostridium perfringens can produce gas gangrene, and there are certain other Clostridia species which can also produce gas gangrene, which you can see over here. For example, Clostridium histolyticum, Clostridium septicum, etc. The predisposing factors for gas gangrene involve accidents, for example, automobile or motorcycle accidents, war wounds, and septic abortions. There are two types of Clostridium perfringens, type A1 and type A2. So we will compare type A1 and type A2 simultaneously. The mode of transmission of type A1 is through entry of this organism into tissues by contamination of traumatized area from foreign particles such as soil or dust, etc. And the mode of transmission of type A2 is again through spores which are in soil, but they can contaminate food, which actually involves meat dishes. Pathogenesis of type A1 involves release of alpha toxin, and you can see that it is lecithinase which damages the cell membranes, including the RBCs, so there can be hemolysis. Then there are certain degradative enzymes which can produce gas in tissues. In case of type A2, there is enterotoxin, and it's same as mode of action of enterotoxin of Staphylococcus aureus. That is, it acts as a super antigen. So there is hypersecretion of fluids in small intestine, and finally loss of fluid and electrolytes. Gas gangrene, also known as myonecrosis, and alternatively you can also call it as necrotizing fasciitis. There is pain, edema, cellulitis in the wound area. Crepitation is a very important finding which indicates presence of gas in tissues. There can be hemolysis and jaundice, blood-tinged exudates, foul-smelling discharge is a very important finding which indicates presence of obligate anaerobic infection. Shock and death can ensue, so the mortality rate is high. Type A2 produces food poisoning. The incubation period is around 8 to 16 hours. There is watery diarrhea with or without vomiting, and actually vomiting is little if it is present, along with cramps and abdominal pain. It resolves in about 1 to 2 days. Lab diagnosis of Clostridium perfringens, day 1. You can take wound exudates, excised or necrosed tissue as a specimen. Robertson cooked medium or thioglycolate broth. Both can be used as an anaerobic transport media as well. Gram staining involves gram-positive rods with spores negative, and there is scanty pus. Why? Because lecithinase damages the neutrophils or WBCs, so there is scanty pus. This is also known as dishwater pus, which is much thinner. Scanty pus and absent spores are suggestive of gas gangrene. You can culture this on Robertson cooked medium and blood agar. Provide anaerobic conditions at 37 degrees centigrade for 48 to 72 hours. On day 2, you will notice double zone of hemolysis. This is first zone and this is the second zone of hemolysis. So there is narrow zone of complete hemolysis and wider incomplete zone of hemolysis. And Robertson cooked medium shows saccharolytic activity, which means that the meat particles have turned pink in color, though they are not digested. So this is saccharolytic activity. On performing gram staining, again gram-positive rods will be observed under microscope. It is catalase negative, non-motile. Go for biochemical tests and sensitivity. Incubate this in an anaerobic incubator at 37 degrees centigrade. And now we will talk about specific tests for identification of Clostridium perfringens. These are specific tests for Clostridium perfringens: Naegele's test, litmus milk test, and reverse chem test. First, I'll discuss this Naegele's test. So, a medium containing 20% human serum or 5% egg yolk agar is taken, and it is divided into two halves. On one side of this plate, anti-alpha toxin is applied, and on this side, there is no antitoxin. Apply the test organism, which is actually Clostridia, so there are lines of clostridial inoculation. Incubate this anaerobically, and you will find zone of opacity. So this is also zone of opacity on this particular site. So this is zone of opacity. And why it is produced? Because lecithinase causes deposition of lipid around the colony, producing zone of opacity. So here there is no zone of opacity. Why? Because this anti-alpha toxin is neutralized by alpha toxin, and therefore there is no zone of opacity. The second test, which is litmus milk test, this test changes color of litmus from blue to red because of acid formation from fermentation of lactose. Acid produced coagulates casein, leading to formation of acid clot, and this clotted milk is disrupted due to vigorous gas production, and this is known as stomy fermentation. Disruption of clot by gas in 24 hours is suggestive of Clostridium perfringens. Reverse chem test. In this case, you will apply group B streptococcus on blood agar, and you will streak Clostridium perfringens perpendicular to it. Incubate this, and you will notice an arrowhead formation, which is towards Clostridium perfringens. And this is because of alpha toxin of Clostridium perfringens interacts with CAM factor of this group B streptococcus, so there is synergistic hemolysis. Day 3, read biochemical reactions. So it is methyl red positive, it produces hydrogen sulfide, ferments certain sugars, and read the sensitivity. Clostridium difficile. It's the most common nosocomial cause of diarrhea. It is an obligate spore-forming gram-positive rod and is present in GIT tract in approximately 3% of general population and up to 30% of hospitalized patients. Most people are not colonized which explains why most people who take antibiotics do not get pseudomembranous colitis. I'll talk about this pseudomembranous colitis in a while. So the mode of transmission is through fecal-oral route, and this is the pathogenesis of Clostridium difficile. Antibiotics such as clindamycin, second, third generation cephalosporins, or use of fluoroquinolones such as ciprofloxacin, levofloxacin. So all of these antibiotics can suppress drug-sensitive members of normal flora, and this allows Clostridium difficile to multiply, and it produces exotoxin A and B. The exotoxin B has more important function. It causes depolymerization of actin, results in loss of cytoskeleton integrity, apoptosis, and death of erythrocytes. So the plaques are formed over the damaged epithelium, and then there is diarrhea. Clostridium difficile produces diarrhea, which is usually not bloody. Neutrophils can be found in stool in about half of the cases. There can be fever and abdominal cramping. Pseudomembranous colitis is an acute inflammatory disease of the colon. In mild cases, this may appear as minimal inflammation or edema of the colonic mucosa. In most severe cases, the mucosa is often covered with these nodular exudates, these raised exudative plaque-like structures. They combine together and they generate an endoscopic appearance of yellowish pseudomembranes lining the colonic mucosa. So they can be enlargement of the colon, which is known as toxic megacolon. There can be bowel perforation and sepsis. Laboratory diagnosis of Clostridium difficile. The first one is enzyme-linked immunosorbent assay test, and this uses known antibody to the exotoxin. So it is rapid but less sensitive. The second one is cytotoxicity test. Human cells in culture are exposed to the exotoxin in stool filtrate, and death of cells is observed. It is more sensitive test, but it is time consuming. Summary: gas gangrene is produced by Clostridium perfringens, and there are also certain other Clostridia which can produce gas gangrene. Clostridium difficile is the most common cause of antibiotic-associated colitis and is typically hospital acquired. Risk factors for Clostridium difficile induced diarrhea includes either extremes of age or prolonged hospital stay or living in a nursing home, or they can be any other severe underlying disease, which is another risk factor. This is your assignment. Thank you.